Hi class, Ms. Turley here. This is Unit G, Part 3, the last part of this unit. And in this unit, we're looking at active transport. So active transport is going to describe for us how substances cross the membrane using energy. So energy, we remember cellular energy is ATP energy, and to get the energy out of ATP, we have to break the bond. So the bonds are going to be in here, and when we break that bond, then the energy is going to be freed up, and that energy then can be utilized to move something against the gradient. So recall back to screencast of this unit, part 2. This is that same gradient, but we're going against it. We're not going down it, so instead of going down a hill, right, we're going up a hill, and not really, right? That's an analogy. That's to help you understand. We wouldn't say that, but the energy is going to be needed to move the item, the substance, against the gradient and into the cell. So some vocabulary words I didn't put in this screencast. I hope you don't miss them too much. First type of active transport is active transport using a carrier protein. So when we talked about the fluid mosaic model, we mentioned 3 functions of proteins. One of them was as a transport, and in the case, of a active transport, we're going to call it a carrier protein. And this word carrier kind of implies that you're being carried. So imagine somebody carrying you across a river, or imagine you fall over and you need to be carried back to the car. This protein is going to carry a molecule across the membrane, and because it's active, it's going to require the input of energy. So generally speaking, smaller particles, and it has to be small enough. The reason it has to be small, it has to fit through this membrane protein. So it's not going to be a huge, large molecule. It's moving against the gradient, so it's going not from high to low, it's going from low to high. So it's moving a particle against the gradient, meaning it's going from an area of low concentration to an area of high concentration. And it must have ATP energy. And we're breaking the bonds in ATP to release the energy and make ADP. An example in the human body would be iodine. Iodine is a trace element that is required for our thyroid gland. Our thyroid gland sits on top of our trachea, and it needs iodine to synthesize a very important hormone we'll talk about next unit, thyroxine. So this is a good example to know. So another pick on the active transport idea, the low concentration, using a carrier protein, breaking the bonds in ATP, energy is freed up, and this is the phosphate that has been removed, and the membrane protein is moving, carrying, pumping, the word is also used, this item from an area of this red, right, item is the substance from an area of low to high. So up the gradient. Second type of active transport is active transport that is going to utilize a vesicle. So it's going to utilize part of the cell membrane. And in the case of coming inside of the cell, endocytosis, so think of if that helps you, the cell membrane is going to wrap around and pinch off and put these items in the case of phagocytosis, it's going to be solid, and this means cell eating, right? So the cell isn't really eating, the cell is engulfing a particle. If it was a unicellular organism such as amoeba, it could be eating, but in the case of our white blood cells, right, our white blood cells roam about our body looking for nasty little bacteria cells that they are going to engulf by phagocytosis. After eating, the cell drinks. So in cell drinking, it pinches off and it takes up some extra cellular fluid. So it's not solid, it's liquid. So the same idea, the cell wraps around and pinches off, but it is liquid. It is not, as in cell eating, something that's solid. And this would form a vesicle with this fluid inside. So just looking again to quickly review, phagocytosis, it's going to be solid and it's going to end up in a food vesicle, food vacuole. The cell membrane pinches off and breaks off. There's the cell membrane here, right? That was there. In pinocytosis, the cell is drinking. So it's engulfing a little bit of the fluid outside of the cell and putting it inside a vesicle. And we don't look at receptor-mediated endocytosis. Both phagocytosis and pinocytosis are examples of endocytosis. Going outside from inside, we are going to call this exocytosis. So remember back, hopefully, from the secretory pathway, and we use the example of a cell making insulin, a cell in the pancreas making insulin, right? We talked about it being synthesized, not with an E, sorry, it being synthesized on the RER, and that is a mess. I'll do that neatly. So there you go. Pancreas cells make insulin. That was back in Unit A. And so if this was insulin, the insulin has to get out of the cell, so the vesicle fuses with the cell membrane and its contents, in this case insulin, would be deposited outside of the cell, exiting the cell. Exocytosis using a vesicle. Like endo, but endo's coming in, exo is going out. So just a refresher, reminder of the secretory pathway, the Golgi body would package, sort, modify the substance into a secretory vesicle. The secretory vesicle would then move to the cell membrane, fuse with the cell membrane, and the contents would be deposited outside of the cell. So that's it for active transport. Can you compare active to passive? This is a comparison question, right? And we see those all the time, right? Describe two types of active transport. So those that use a carrier protein and using vesicles. And remember, using vesicles, there's two kinds, going in, endo, going out, exo. Can you give examples in the human body? And this is carrier. I will make an effort to try to be neater. Can you give examples in the human body for each type of active transport? And most importantly, if you have questions, write them down right now. Go back and see if you can answer them. And if not, bring those questions with you to class and ask me, right? Please. And thank you. Hope that helped.